Okay, let's get started. So welcome to the Need for Speed seminar series. Today we have Stephen Bulpart, who's going to be presenting. He's actually the second MD that's part of our series, and also the second MD PhD. He got his MD from Harvard Medical School and his PhD from MIT. A pair of places for which I know no better place to get an MD and a PhD. He has been serving on the as a professor in the Electrical and Computer Engineering Department since 1999. He holds appointments in the Bioengineering Department, the University of Illinois College of Medicine, and he is the head of the Biophotonics Imaging Laboratory in Beckman Institute. If that wasn't enough, from 2006 to 2008, he served as the founding director of the Mills Breast Cancer Institute and has held a joint position with the Carle Foundation Hospital and the Carle Clinic Association in Urbana. And I guess in his spare time, he's been doing some optical imaging, and that's what we have him here to talk about. So thank you, Stephen, and we're looking forward to your talk.
Well, thank you very much, and I really appreciate these opportunities to come and share with all of you and others what we've been doing both here and at Carle, and kind of crossing this cultural divide between engineering and medicine. And what I want to emphasize today is really so much about not only the work that we've been doing, but where it can go and where we need help, and hence the need for speed in what we're doing. You'll, I think, quickly see that in our case, what I'm emphasizing is volumetric microscopy. That if you imagine that we now want to be looking at tissue at the micron level, where cells and molecules reside, rather than anatomy and structure, then we very quickly have very large data sets if we're talking about whole organs or tissues. And you divide that up in cubic microns, and you have a lot of those. And we have to be able to do processing kind of at that scale. So I always start by talking about this concept of translational research and technology. People have different definitions of what translational research is all about. But it's essentially fairly broad and encompassing of what we do. It may be that basic biology benchtop research. It may be the engineering lab, the technology that's being translated. It may just even be virtual and computational. And it's really trying to interface that and look to see where technology is and can be used in the clinic. And you don't have to look very far, and you see that there's technology everywhere. And you don't have to look much further than that and see where technology really could still make an impact. And so much of what this is about is almost a cliche, bench to bedside, but also back again, that connecting the people that really are the users in the clinical world and how they can further improve and provide us the questions that engineers and scientists need to address from the bottom up. The other bit of motivation for this type of work is that NIH and others realize that the magnitude to translate findings from the research realm into practice has been grossly underestimated. And they put out some numbers that it currently takes an estimated average of 17 years for only 14% of new scientific discoveries to really enter day-to-day clinical practice. There's a whole lot of reasons this exists, even as much as socioeconomic, political, bureaucratic, but even, we've got great technology, we've got perfect applications. The bottleneck really is somewhere in between. And that's where I think translational research is about, is trying to work on those bottlenecks and how we can actually reduce that. To give a brief overview, Sanjay mentioned my Biophotonics Imaging Lab at the Beckman Institute. I wanted to give an overview of that. We do a lot of different things, starting with optical detection and imaging systems. We design portable systems that we take into the clinic. We design even new optical laser sources. And then we also engineer how we can get this light to the tissue through microscopes, through catheters, through devices that allow us to bring that to the tissue. We also, every bit of this, the digital acquisition, the image and signal processing, the display, the feedback systems, all of that is kind of rolled into this cart in this project. Coupled with that is really the software and the computational aspects that we're interested in. And primarily it's because we're interested in extracting the most clinically and diagnostically useful information out of those optical signals that we can measure. And so there's a number of different ways that we can analyze this, process this. You know, we want to make sure we're getting the most bang for our buck. And that really is a whole other realm of improving the data that we acquire. With these systems, then, we can start at the small scale and work up. We have systems that look at the cellular level, microscopy systems to look at tumor cells, developmental biology, engineered tissues. And on a little bit larger scale, we can apply these systems and look at very high-resolution structural and functional imaging. This happens to be a tadpole that's about 1 millimeter or 2 long. This is a millimeter-sized tumor in a dog muscle. Very high resolution. We can also look at functional properties like Doppler flow and others. To get down even to the subcellular level, we've developed molecular imaging techniques. These are primarily contrast agents that are engineered to be targeted to certain tumor cells or cells of interest. This is an example of a microsphere developed with Professor Ken Susslich in chemistry here. And these are fairly large, a few microns. They can carry drugs and deliver drugs to the site, provide contrast and also therapy. These are magnetic nanoparticles to provide contrast therapy, also multimodal with MRI, and gold nanoparticles and others. And then finally, we can take these systems and these agents, we can do this in cell culture, in small animal models, preclinical models, in human tissue, and then eventually even in human patient trials. Mostly what's at this stage is just using an imaging technique to look at the endogenous or inherent contrast from tissue. We really are not approved to do any of these more experimental agents. But much of the clinical studies, which I'll talk about today, are just simply looking at some of these advanced imaging techniques on clinical patients. So the primary technology is called optical coherence tomography, or OCT. This was developed while I was at MIT in Jim Fujimoto's lab there. And it really is an optical analog to ultrasound. So instead of looking at sound waves that get reflected off, we look at near-infrared waves. And we get very high resolution that we can then scan over areas of tissue to get three-dimensional volumes. These happen to be cells in a tadpole. Or we can do temporal acquisition, stay fixed at one plane but acquire over time. And this is the beating heart of that tadpole. We can see blood flow in these arteries and through the heart. And very fast acquisition, that cellular level resolution, volumetric imaging. This is all digital data, so we can do all sorts of processing on this. And really we're after, can we use this information for feedback for these procedures? If we look at the heart of what an OCT system is composed of, a lot of it is fiber optics. And in fact, we borrow a lot of the same technology from telecom. And as telecom has advanced, we've leveraged that. And we've used their advanced detectors and optical sources and fibers and couplers and really refined these systems over the last decade and a half. So primarily it involves an optical source, some fiber optic couplers. This is an interferometer, so we compare a sample arm with a reference arm, and then we have different types of spectral detection systems. This system can be very compact. This is on a medical ultrasound or an endoscopy cart, but it still can be a factor of maybe 4 to 5 times smaller than this. We interface then this to optical systems, and in medicine there's actually quite a lot of optical systems in place. There's surgical microscopes, there's different types of probes, there's endoscopes, different cystoscopes for looking inside the body. And we can interface this new type of optical imaging through a lot of those same systems. So how do we do this? Well, we will scan our reference arm in the most simplest case, and that produces means that we can measure the amount of scatter coming back at a different depth in tissue along one point. And this then allows us to collect what we call an A-scan, or a depth scan, that represents the amount of optical scattering versus depth. This then could be digitized and a grayscale assigned to that. Then to produce an image, we simply step our beam over, or we translate our sample, and we can collect a 2-dimensional image or even a 3-dimensional volume then of that data, and just really repeating this process of these depth scans. We can also, in another mode, do en face imaging, which is much like standard microscopy, where you pick a plane, you have a very tight focused beam, and we can collect an image in this en face plane, and then move our focus up and down to collect that 3-dimensional volume that way as well. this technology has really taken off, and in fact, the first publication, it was in 1991. In recent years, a lot of this has been fueled by applications in ophthalmology. The first application for OCT was in the eye. There's now really 10 to 20 different commercial vendors, Zeiss and others that are making commercial medical instruments for the eye. And in fact, I always like to tell the story. I was over at Carl Clinic and I had to see an ophthalmologist there, and I was checking in, and something caught my eye on the check-in station. And the nurse saw me looking, and so she slammed the chart shut. It was a patient's chart, and thought I was prying on patient data. But what caught my eye was an OCT image in a patient's chart. And I asked if they had an OCT system. I wasn't aware of that. And sure enough, they do. And the nurse, you know, I was excited about that and was asking the nurse all kinds of questions, and she couldn't answer me. And I asked the physician, and really it was surprising, I guess, that there's two messages. One is that it's nice to see this technology really getting to the level of these community hospitals, and that was good to see. The second was that, you know, here just across the street is an OCT system, and I wasn't even aware of that. So there's a disconnect even locally between what's going on, I think, technologically at the hospital and what's going on here at the university. So we hope that this type of upswing is going to really further this technology for many different applications across biology and medicine. If we look at breast cancer in particular, we can see that there's lots of uses of optics in breast cancer. You know, the pathology lab is traditionally known for using optics in their microscopes. And in fact, light microscopy, fluorescence microscopy, those are really been standards for decades, for centuries. More recently, there's use of optics for looking at optical mammography, and light can actually go tens of centimeters through tissue. When it goes that deep and that far, it scatters around a lot, and it's more as diffusing through tissue, so you lose spatial resolution. So it's very good at looking at functional parameters, hemoglobin concentration, blood saturation, scattering, but the resolution is still on the order of millimeters, not the microns that we can get with optical coherence tomography. So that's where we come in. Coherence imaging is really about training depth of penetration for high resolution and being able to reconstruct these images that look very much like histology. So if we look at where patient flow is through the standard of care, we see that patients are screened with mammography. If there's abnormalities, they may undergo a needle biopsy or open surgery. There's a gross pathological exam, microscopic exam. And these are the usual players and technologies that are currently used. And if we look at where optics can come in, it really potentially can come in at each of these stages. And I mentioned optical mammography, but we really, with coherence imaging, want to focus on some of these latter four areas: needle guidance, margin assessment, lymph node targeting, and sampling guidance. So really, what we're after is to change this paradigm for diagnosis, that we want to move where diagnosis always occurs in the path lab and move it to the point of care, move it to real time, where that information could potentially be used for feedback back to the surgeon or the physician and make decisions there. Now, this data acquired here in real time may not be equivalent to the standard of care or the gold standard, which is histopathology. I don't think we're ever going to replace the pathologist, which is probably a good news. But I think it's going to help us. It's going to help the pathologist because it's going to help provide information in real time to make those decisions. So we translated this technology and have been performing and just now finishing up 3 clinical studies that were in conjunction with Carl. The first is intraoperative assessment of surgical tumor margins for assessing lymph node status and for optical guidance of these needle biopsies. And I'll explain and go through each of these. So if we look at surgical treatment of breast cancer, there are certain interventions that are done. The norm is becoming lumpectomy. So if a mass is identified, diagnosed as cancer, a lumpectomy, that mass is resected out, as opposed to mastectomy, although there's still a number of mastectomies performed. They also do what's called a lymph node biopsy, and I'll explain that in the next couple slides. But it's essentially trying to stage the disease. Has the disease spread from that primary tumor? And we want to ask ourselves is, can this real-time intraoperative optical imaging really provide us with accurate information to make decisions? Well, where do we want to use this? First of all, survey surgical margins. The margin status, when a mass is resected, you can see that the tumor is really in the center, and that may be about 1 centimeter in size. But they resect a mass that's 5 centimeters or larger because they want to try to get as much of these cells that may be spreading away from that tumor resected. But they have no microscopic way of making sure that these margins are really clean unless they send that to pathology. And in fact, making sure that those are clean is really the most important factor that governs local recurrence of a tumor there. Now, the typical positive margin rate in these procedures today, it varies. It can be as low as 1%. It can be up to maybe 12%. If you actually look at every square micron on one of these margins, studies have found that over 50% of these margins have positive tumor cells left behind. So it's a significant amount, and it raises this question, which I'll point to in a second. Secondly, we want to assess this lymph node status. So lymph nodes really are the filters of the body. They collect drainage from, say, the breast or the tumor. If tumor cells are spreading, they may filter out and collect there. It's used for staging. Well, only 25% of those lymph nodes that are resected contain metastatic disease. So we're resecting far too many of these that are negative. And in fact, the decision point for these lymph nodes is really binary. Is there metastatic disease or is there not? So it's a rather binary decision. If we take too much out, the problem is you disrupt the lymphatic drainage of the limb or of the tissue, and you get fluid that accumulates. And this is a very painful, it's a morbid condition, lifelong, and it's problematic. So if there's a way of assessing, making that binary decision in the operating room, yes or no, that I think is what mostly we need to know. Both of these speak to the fact that, and as engineers we can appreciate, we need to improve sampling rates. Oftentimes, because of time and cost, we undersample that tissue. So there's only a discrete number of sites that are visually assessed by the pathologist that are actually sent for microscopic exam. And we don't know what is being missed. Obviously, if you look everywhere, you see that there's a far greater number of sites that have perhaps positive margins. So this is what optics, what we intend to kind of address, is point-of-care assessment in real time and also improve the sampling rate of the tissue that we're looking at. So we started this using a rat mammary tumor model that sort of recapitulates the progression of disease in the human breast. And just looking structurally, we see in the normal mammary gland, it's a lot of adipocytes, fat cells, and those show up as low scattering. If you've got tumor, you have a high cell density of nuclei. They're highly scattering, and it looks very different optically and based on scattering. If we look in this particular model, it begins with sort of hyperplasia ducts. So an increasing number of cells in these duct structures. And again, they show up as scattering areas against the low scattering fat and adipocytes. If we look in longitudinal section, we can see that here as well. So that's an essential message to interpret these images, is more cells, dense cells, high scattering, darker, they show up against this background. And really what we're after is, can we identify these nests of potential tumor cells that have been migrating away from this tumor? We see very good relations and correlations between our images and that histology. This is a human invasive ductal carcinoma. And we also see it for these lymph nodes. And in this case, the scattering plays a role as well. In normal lymph nodes, this is a 3D image, we see fairly low scattering in this cortex. When there's metastatic cells, it changes the architecture and the scattering of these particular cells. Yeah, John? Yeah, so the question for our remote viewers is what really is changing the optical scattering? And it really is kind of all the above. So these cells, with a higher density of cells, you get more nuclei. The primary scatterers within cells are the nuclei, mitochondria, and oftentimes the lipid membranes. And so these represent a change in the optical index of refraction. They set up and be scatterers. And so when you have that change occur, you see more increased scattering over this. Now, you also see that there's lots of cells here. So the scattering, optical scattering that occurs in tissue is rather complex, because when you have scatters the size of the wavelength, on the order of the wavelength of light, those are called what's called me-scattering. And oftentimes that can be forward scattering or backward scattering with very small changes in their sizes. And so when you have a disruption of that architecture, you start to see, yes, maybe not so much higher density of cells, but a change in the spacing, a change in the contrast between the scatters and maybe other cytoplasm. And that's what also contributes to that type of increased scattering. So there's lots of different factors that will contribute to that. Unfortunately, why breast and lung and other tissues work well for this is they have a fairly low scattering background, and we're really detecting changes from that. So we made our systems portable. We translated them into our clinic. That's why we put on our white coats, because it's now clinical imaging. And we have a research protocol that's in place that allows us to bring our instrument into the operating room and to be stationed there to receive that lumpectomy specimen as it's resected out from the patient. Now, we're in the operating room, but we're still, in this instance, working with resected tissue because we want to minimally interfere with the standard of care. We get roughly 5 to 10 minutes to analyze these margins, and then we hand this back to the regular team. We will scan the margins. We will mark areas that look suspicious and which look normal to us. We may do needle biopsies, which is shown here, and this then is sent to pathology. Pathology then will examine the tissue. They will take their own sections, but also section where we found something that looked interesting. And then, of course, we can compare our images with the histology. Now, our end goal, of course, is to have essentially like a laser pointer that you give to the surgeon, and when the surgeon is in looking at the tumor cavity or identifying tissue, then he or she can make that assessment at that point as well. We did a training set of images on about 17 patients, and this is what we saw. We saw a lot of normal margins, which is very good. It's primarily composed of these fat cells. We see some scattering areas. If we have surface blood, which is an artifact, we see that as a dense scattering. Red blood cells are very highly scattering. That can be washed away. Cautery, which is often used to cut out the tumors and coagulate vessels, that shows up as a dense scatter, but fairly superficial. And then when we start seeing heterogeneous areas, these are more indicative of tumor, and a more invasive tumor if they run deep. And so then we did our study, which involved about 20 patients, and again, a lot of normal tissue, which is great, but we did find intraoperatively positive tumor margins, and even very small isolated foci of tumor cells. This happens to be about 1 millimeter in size, confirmed with histopathology, but this is very promising to us because this is real-time data, just one image out of gigabytes of data, and that raises these questions for our need for speed, is that how can you physically go through this data with the naked eye and being able to do this type of identification. This is also frustrating for a lot of my students because we're finding positive margins, and we can't inform the surgeon about this because these are experimental techniques. We can't modify the standard of care. It's not approved for that. So we mark these sites, pathology finds these sites, and the patients have to come back for a second surgery because there's positive margins. So it's very frustrating at this stage, but it's hopeful the fact that we really are finding these positive correlations. And these are just other examples. These are different images through a 3D volume. This is invasive papillary carcinoma confirmed with different types of immunohistochemistry staining and standard H&E staining. And like I said, we can do 3D volumes, so we get three-dimensional data sets that we can then analyze the scattering at all these different angles. We can computationally section this at arbitrary angles, which is very different from what pathology can do. They usually have to pick a single plane and section along that plane. And so there's a lot of flexibility here. These results, you can see 38 patients, a wide variety of different types of cancers, ductal carcinoma, invasive lobular carcinoma, mammary carcinoma, and of these we're getting numbers that are very good, a sensitivity of 100%, a specificity of 82%. Very hopeful and suggestive results. Okay, the second type of imaging and project, as I mentioned, was this intraoperative lymph node assessment. And for this, we really want to be able to look at and image these lymph nodes. As I said, the breast tissue often will drain to these lymph nodes in the armpit or in the axillary, and this is where surgeons will actually dissect these out. Now, the reason for this is because, from a scientific point of view, this is a very interesting structure because we believe that as much of the metastases that occur through the bloodstream also occur through the lymphatics. And I think we don't know enough about, I think we underappreciate the lymphatics as a way of spreading disease. There's even a lot of strong selective forces on these tumor cells to sort of take up residence in these lymph nodes. And some people have even thought, do these serve as kind of a bridgehead where tumor cells can reside and develop and then become more metastatic before they move on and spread. So there's a lot of interesting physiology in these lymph nodes. They also work, these lymph nodes work as filters, and so this afferent, this incoming lymphatic fluid will enter and will be filtered within these cells here. This is filled with a lot of immune cells to identify foreign particles, viruses, bacteria, tumor cells the best they can, and basically address those from an immune response. However, what's important is that looking at normal, reactive, and metastatic, there are morphological changes that occur in these tissues, and this is what we hope to detect intraoperatively. Reactive lymph nodes, you can see that you get a lot more of these little follicles, these focal regions, which are responses to some foreign body. You can have a reactive node just if people get an infection or bacteria. Maybe some people have swollen lymph nodes in their neck. That doesn't imply that what's causing the reaction is a tumor cell. But if it is a tumor cell, it may go on to become a metastatic lymph node. And this is part of the diagnostic challenge that we face, is trying to differentiate those. Now, I mentioned briefly that sentinel lymph node process. This is done by accepting the hypothesis that you have a primary tumor that will drain the lymph from that. And first, there's a sentinel node. There's the primary node where that fluid collects. And then it goes on to other nodes. And so if you can find that primary node, that sentinel node, and determine if there's tumor cells there, it's likely the tumor has spread. If that's negative, it's likely the tumor has not spread to that point. So right now what they do is they inject radioactive agents, technetium-99 or blue dye, and they inject these at the site of the primary tumor, and then they look intraoperatively to see, what has lit up. When they go in intraoperatively, they find, it's hard to see it, but there's a little blue lymph node right in there. They look for that. And so all this tells you is what is that first sentinel node. It doesn't tell you if there's metastatic disease or not. The idea is to find that node, to resect it, and then send it to histopathology. But of course, there's lots of problems in making sure that that's the lymph node, the sentinel node. So for our studies, we started with rat. Again, back to our rat model. We were looking at identifying these follicles and how they correlate to histology, trying to look at the scattering features within this cortex for these normal lymph nodes. And then when there's metastatic disease, and these happen to be human nodes, we see again a very distinct change in the architecture of those lymph nodes. What we did for this feasibility study was look intraoperatively at 16 patients. Some of these were the same patients in our tumor margin studies, and looking at these lymph nodes that are resected. Now this fact points to one of these problems, and that is of these 16 patients, 12 of those nodes that we looked at were normal. So in a sense, 12 of those really didn't have to be taken out. Perhaps none of these had to be removed if we could have made that decision. Only 3 of them were metastatic, and 1 was judged as reactive. So when we started doing this, we first had to make sure that the dye that's injected did not interfere with the light we were using. And in fact, this sort of mahogany absorption peak is the absorption for the dye that they use. And we're out here at this wavelength, so far from that absorption. So the dye that's present doesn't interfere with our signal that we're collecting. These are some 3D cutaways of these lymph nodes, and this again was acquired by pointing the beam from the outer surface and looking in. And we can see that we see this capsule, we see some of the cortex that's inside here. And again, this is data that we can look at 3-dimensionally. So part of our challenge now is to say, can we differentiate normal from reactive from metastatic? A lot of it has to do with the structures that are present. What we found is that when you get a reactive node or a node that's metastatic, a lot of the capsule area becomes effaced. So we no longer can find a very identified capsule where the lymph would flow through because that lymph node's becoming enlarged. And when it becomes metastatic, we know that this cortex, this scattering here, looks very different from the more homogeneous scattering of a reactive node. So this is still early, but a lot of us, we have to do some signal processing to analyze this. Well, the last set of procedures that we've been looking at is related to optical biopsies. So you'll recall that in that flow chart for patient care, one of the very first things that we have to do is determine, is that mass found on mammography, is that mass really a tumor or not? And so to do that, you have to get tissue. And you have to use what's called a biopsy needle or a biopsy device to stick a needle in there and either take some fluid out and hope you get some tumor cells, or take a core needle biopsy, which is literally a core of tissue that preserves the structure of the tissue, and then look at that under a microscope to determine what pathology you have. So again, we ask the same question: can this resolution be used, this imaging be used to guide these procedures? Well, the problem is, with these fine and core needle biopsies, you're trying to essentially hit a very small lesion deep within the breast and do that with what I think is rather crude guidance tools. People will use ultrasound, or they'll use what's called an X-ray stereotactic apparatus, which does X-rays from two different orthogonal directions. And you're trying to hit a small lesion within a three-dimensional volume. And oftentimes you're trying to hit areas that are just really microscopic. So this is a challenge, even for experienced people. And part of the problem is that you're trying to image and guide using modalities that are external to the tissue. And our approach is to say, let's use some sort of embedded optical needle. So we're actually sampling at the tip of the needle rather than trying to sample from the outside. What's called non-diagnostic sampling, these rates, depending on the experience, can be up to 15% or so. What that means is that you've got a suspicious mass on mammography, but every time you put in a needle to try to get tissue out, it comes back as normal tissue. And in those cases, you generally have to go to the operating room. So this really allows us to assess mass and tissue in real time, perhaps for more accurate wire placement. And it comes back to just, we want to sample more in real time and guide these procedures. So with that, my group's been developing different optical needles. These are single mode fibers within a needle barrel, and we can do all kinds of designs. We can look out the side here, we can look out forward. This is a rather crude prototype that spins the needle and also can retract the needle, so we can get a cylindrical three-dimensional volume of this tissue, all at the very small size scale. We can also look at things such as refractive index. As was mentioned, refractive index can help us distinguish between normal and tumor. And these are different designs that allow us to measure the refractive index of tissue. And some of these will fit then into these core biopsy needle systems. What we recognize, and where computational assessment now plays a major role, is that we want to differentiate between tissue types. So in these cases, we're not imaging. We're taking axial scans, a single depth scan, and we want to ask, can a single depth scan tell us enough about the tissue to make an assessment of the tissue type? Well, the answer, we think, is yes. If you just look at these depth scans for the different tissue types, fat, adipose, tumor, or stroma, which is a lot of the connective tissue, you see that there's axial scans look different just by our eyes. We see discrete peaks here, which correspond to membranes. There's wide spacing, gradual attenuation. The tumor has few discrete peaks, a lot more rapid attenuation because this packed density of cells, and stroma is somewhere in between. And so just if we can do this by eye, of course, we can train a computer algorithm to do automated tissue type classification. And so using signal processing, fairly crude techniques looking at just the mean distance between these peaks in scattering, if we look at the spatial frequency of those axial scans, we can begin to differentiate these. And just like we do for various spectrum analyzers, we can look at the spatial frequency content for each of these types of scans. And we can use these features to begin to differentiate tissue types. Well, why is this important? Well, you can start to see, as I mentioned, if we are looking at three-dimensional volumes at micron scale, there's just no way that anyone can analyze these signals in real time. And so we have to have some automated way or computer-aided detection or diagnosis to give us clues. So I was showing you single depth scans, but also remember, these images are made up of columns of these depth scans. And so we can do the same type of analysis on any one of our images, and we can let the computer begin to identify and classify different tissue types based on the spatial frequency analysis or other features. If we have images, of course, we can begin looking at texture analysis within the images themselves. And we've got this multidimensional data set to then classify these tissue types. When we do this, our sensitivity and specificity get very good. So if we look at 5 adjacent scan lines, we can get 99% sensitivity, 87% specificity. Maybe not everybody knows what these refer to. Sensitivity is what is your percentage or likelihood of finding something positive against all the ones that you know are positive. Specificity is the likelihood of you calling something negative against all the possible negative specimens. And so really you want those as high as possible, but those are very good. In fact, if you compare to other modalities, mammography and ultrasound, we're doing quite well compared to those. And yet this is something that we can achieve at the point of care in real time, say at the tip of a needle. So our next stage then, that was all in animal tissue. We now use our same cart. We have our needles that are brought into the operating room, and we have a calibrated needle. On our specimens, we insert those needles, and we know given depths into the tumors. And for the most part, this is a lot of normal tissue out here, and we want to see when we get to really the core of the tumor. This is emulating what would be done in the outpatient clinic when a person comes in just to get a needle biopsy. And then, of course, we do histology, and we correlate where we think that needle tip was located, what histology calls that tissue to be, and we compare it to what our data showed. And this is just an example of 10 different cases. The asterisks mark where we think that needle tip was located, and just some examples of the different spatial and spectral features that we can identify in these tissues. The study results, this is just to summarize 9 patients. If we look at combining the scattering profile and refractive index, our sensitivity and specificities are up there. They're not as high as we would like. We can trade off, do some statistical analysis to boost these up into the 90s. But if we look at some of these features alone, like scattering alone, we don't get very good numbers. And we really think that this is because it's difficult to register where we were at in the operating room. By the time we hand the tissue over, it gets distorted and squashed and processed, and finally to the pathologist lab. We think there's lots of errors in terms of positioning. And so if we just compare what we were getting in the lab, where it's much more controlled, we think that these numbers actually are more realistic to where this type of diagnostics will be. So the last part I want to mention is some work we've been doing with Professor Scott Carney and others, really using some of his theoretical mathematical framework for inverse scattering and how that relates to optical microscopy. The reason we want to do this is because we need a fast way of collecting microscopic data over entire volumes. Instead of this type of point scanning that we have to do, and we have to do this very quickly, are there other ways to capture that same data? So this idea of inverse scattering is really the inference of the properties of an inhomogeneous medium from the waves that we can measure that are scattered by that medium. And we ask, how can optical microscopy then be improved by this method? So if we only have to look to other areas in medical imaging to find the impact of inverse problems. If we look at radar, if we look at X-ray projections, NMR spectroscopy, and up till now what I was telling you about OCT, these all have advanced technologies that use this inverse scattering. And these have evolved into synthetic aperture radar, CT, X-ray CT, MRI, and what we call interferometric synthetic aperture microscopy, or ISAM. How ISAM works is, if you think about SAR, synthetic aperture radar, you're basically collecting, you're creating synthetic aperture. You're collecting reflectance data from one particular angle. You're flying over a scene, collecting the same type of information from multiple angles, and you reconstruct that to produce that image. Well, if you think about what we're doing in microscopy, we're really doing something very similar. If this is our Gaussian beam, and this is our focal plane, and we have scatterers, which represent cells or nuclei within that tissue, as we scan our Gaussian beam across, you'll see that these scatterers out here are actually being illuminated by different plane waves, optical plane waves, if you decompose that Gaussian beam into different plane waves, and at different angles. And so what we see in our image currently is that this cell that happens to be near the focus looks like a cell, but things that are out of focus tend to be blurred. Okay? But if we know the phase, and we know and can reconstruct what these wavefronts look like, then we can actually reconstruct what that object is. Instead of that blurred object, we can actually identify and reconstruct that as a point. So this is sort of a resampling in Fourier space, and it's really a topic of a whole other lecture. But it's essentially resampling to go from that acquired coordinate system into the object space, which is what we're most interested in. And these are examples of this. So in standard OCT, if we look at the focal plane, we see that these are point scatters, and they look relatively in focus. But as we move further and further away, we see this blurring effect. We even see interference from multiple scattering. These coherent waves scattering off adjacent scatterers, they create interference. But when you apply this algorithm, what happens is that you basically can reconstruct the object structure. And you can see at each of these planes now, we see these point scatterers as the object truly is. And so this allows us to go from this sort of image where cells are in focus only at the focal, within the confocal parameter, and blurry out here, to really spatially invariant resolution. So just by a single illuminated area, we can now collect microscopic in-focus data throughout that entire illuminated volume. And that's powerful. We started doing this with breast tissue, and you can see this is histology. This is kind of the raw data, and as we do ISAM correction, we can really clean up and improve all of these structures to look and resemble very closely what the histology shows. And so, and this is the fact that we can actually do some of this in real time as well. This starts to get complicated because as the tissue gets more highly scattering, we don't know really what the limits are as to how well ISAM can reconstruct and improve sort out all that multiple scattering that's going on, and that's kind of an active area of investigation. But what the potential of this is for us to very rapidly be able to acquire microscopic data over volumes, and hence even more the need for speed for doing this processing, for analyzing this data for tissue type classification. I just wanted to mention and acknowledge really the large number of people that kind of contribute to this. There's people from many different disciplines: engineering, physics, computer science, immunologists, biologists, a lot of our clinical collaborators that really allow us to really take part in the patient care over at Carle. Other people not mentioned here, obviously Scott Carney, Dan Marks, Tyler Ralston, a lot of other former graduate students that were working on ISAM in the past. And so I think all of this is really what's necessary for this interdisciplinary, for this translation from this technology into the clinic. And I just put this up if people are interested. We've got a lot of other different projects in our biophotonics imaging lab, and all of our publications can be downloaded as well on ISAM or on OCT. So I thank you very much for your attention. I hope that this at least provides kind of a framework and maybe the problem which you can perhaps help with. The fact that we need to do this type of processing fast, we need to do this in a parallel fashion. We have to be able to do image processing, tissue type classification, all in real time, and provide that information back to the surgeon to make those decisions in real time. Thanks.
Okay, we have time for a few questions here.
Yes, we may.
So for ISAM, what is the alternative ISAM
Yeah.
Do you need to prepare the tissue into thin slices, or what's the process, and how long does it take to go through that process versus
Yeah.
ISAM?
Yeah, so what ISAM does is it eliminates a lot of the mechanical scanning that we would necessarily have to do. You can imagine if we take a cross-sectional image, there is a focal point within that image, and as we go for higher and higher resolution, that focal region gets smaller. And so that means there's a lot more that's out of focus that traditionally people have just thrown away because it's blurred, it's out of focus. So what ISAM does is then we can then capture an image at one plane. ISAM will then allow us to make that entire illuminated plane in focus and usable. So we don't have to have the mechanics or repeat our scans at each micron depth to get that volume.
So mechanics meaning mechanically shifting the device.
Correct. Yeah, shifting that, controlling for that with high precision, that's a problem. The other, we've explored a lot of other geometries where we can do this in full field. So instead of point scanning and collecting a single point and building up an image point by point, it is possible to illuminate the entire tissue with a wide field beam and do this ISAM reconstruction in the full field. There's lots of other technical problems with that, but potentially in a snapshot then you can collect a three-dimensional volumetric image at micron resolution. Yeah.
I don't know how you actually collect this data, but it seems like you need very high precision mechanical pointing of your laser beam, of your beam. Do you think that can be alleviated with increased computational power?
Yeah, that's a good question. So there's lots of different ways, our beam delivery systems, to acquire this. For the examples I gave, we really had just a galvanometer that will direct a beam, scan a beam across the tissue. And you can do that at very high frame rates. You can have it XY, so you can scan over an area, and then OCT gives you the volume in depth. And then you can manipulate the sample underneath to move that volume around. There are also handheld probes that will have a scanning mechanism in there. But what you mentioned is actually one of our current projects, and that is we want to apply cross-correlation algorithms that say, let's say instead of worrying about mechanics to scan, let's just have a probe that has a single beam coming out, and we just simply by hand move this beam across. And by looking at the data that's acquired and looking at the cross-correlation between adjacent scans, we can say, are we at the same point or did we move? And if we moved, that represents a new column in our image. So you have an acquired array, and you process that and then extract really what the image, you essentially either average out or you avoid the redundant A scans. And so that's kind of an active area that we're looking at. Another, in fact, area that speaks to this surgical guidance technique is to use the tissue type classification, but then code that into an audio signal. So a surgeon, is going to be scanning and primarily is looking at the tissue. Doesn't have time to look at monitors or look at pathology. But in a sense, if that classification could then identify suspect areas and even ring a bell, sound a buzzer, light a light, can provide some audio signal, much like a Geiger counter would. I think that's another add-on that we've been investigating to computationally analyze this data. There was a question behind you here.
I'm just wondering where the deep parallel process and how long do the algorithms take now?
Yeah. So if we look at the two different modes, OCT and ISAM, as being two discrete things, right now for OCT, we really are limited computationally because the rate at which we can scan these A scans in our systems now at about 30,000 A scans per second. And so that's 30,000 depth scans per second. You can break that up if you have an image, 500 columns across, that's 60 frames per second, or you can divide that up into a volume of data. Each one of those depth scans, I should say, I didn't go into a lot of the scanning techniques for OCT, but one method is to capture a spectrum of data coming from that depth. We have to do an inverse FFT. So that is kind of the process, the bottleneck that has to be done on each A scan. And then that will allow us to look at the intensity profile in depth. That then is assigned or put into an array that will represent that image. There's a lot of other processing, dispersion compensation that can be done. On top of that would be your classification. Now with ISAM, that same process, the changes that have to happen in ISAM really is to ensure phase stability, and you have to deal with a lot more of the, you have to kind of oversample the tissue a little bit more in order to add back to that ISAM algorithm. We have shown the videos that we're showing there was ISAM done in real time. So we are able to go at about 2 frames per second with that ISAM processing. So a factor of about maybe 8 times faster than, well, 8 times slower than OCT. So I think there's still kind of a need for us. We really want to get to large volumes of imaging because, again, all these areas that we're looking at are on the millimeter scale, and those tumor margins or those lymph nodes are on the tens of centimeters scale. And so we need very fast ways of capturing all that.
Steve, let me quickly interrupt here and say that if anybody's watching online, you want to ask Stephen a question, you can type it in on the browser window. So I'll let you do that. And I think there was another question here. We'll take a few more here.
Okay.
And then go back
Okay.
online.
Yes.
Just follow up on the previous question. It sounded like FFT was the bottleneck for OCT, and Fourier transform was the bottleneck for the first algorithm, the OCT. Is it also the bottleneck for the ISAM?
Yeah, so everything we have to do for ISAM, we have to first do for OCT. So ISAM is essentially OCT, but keeping better track of the phase information of the light and sort of oversampling in OCT, and then using that oversampling to basically reconstruct the ISAM image. So there's just another level of the processing that has to go on for the ISAM. I think a lot of these, and again, I think a lot of this OCT processing, people know that you can do FFTs, at tens of thousands of these per second, and you have dedicated cards. And I don't think that's enough. I mean, from what we're looking at, we really need to be able to, in some way, the best we can, parallelize all this, so we can look at these volumes of tissue.
By parallelize all of this, you mean make the FFT faster, or do things in addition to the FFT?
I think in addition to the FFT, or basically split it up among parallel processors, and even if we look at more of the volumetric imaging, where we're no longer talking about single columns, but we're looking at this volume of data that has to sort of have 3-dimensional spatial Fourier transforms, and so I think that's where we're really quickly going to run into some issues. It used to be, when this all started, that we were limited by the optics, and that we couldn't scan fast enough. The PCs were fine. We did, early on, we used like an FPGA implementation to do a lot of the filtering of these signals. A lot of the PC processors, caught up, and those are what we've been using since. But we really, what I'm most interested in is how we can use a lot of these, these GPUs and other methods to really kind of carry this to the next step. Yes? So the light frequencies, perhaps the best is, I think maybe wavelengths. So the wavelengths of light, we typically will work in what's called this biological window. And so that's from 700 nanometers up to about 1300 nanometers. And we choose those depending on the optical properties of the tissue. 1300 nanometer light is outside of the range of most, like silicon CCDs. You have to use in-gas arrays for detection. It's beyond our visual responsiveness of our retina, but that is really affords the best penetration. It's low scattering in tissue, and so that is the wavelength. You can calculate the frequency of what that would be, but when we use an interferometer, what's unique about interferometry is that when we split the light into a sample arm and a reference arm, and essentially we're then looking to beat the signal from both arms. And what we detect then is the beat frequency between those two. And that's where we can get information about Doppler changes of moving scatters. It's where we can get a lot of the spectroscopic content a lot of other features out of the light from that process. Yes?
In OCT, is it a matter of reconstructing a large number of FFT images, or is also a matter so that you can use human eyes to look at them, or you also need to have some mechanism to classify these images and then help the human to zoom into the right area?
Yeah, it's both. I think that I went through a lot of different areas that we need help in. The first is just capturing that volume of data, and in general in surgery, we run into this limitation, and it's common through many different applications, that you really have a microscopic or a small field of view that you're sampling, but you need to sample over a large area. And how do you do that? I think there's three methods. One is you use multimodal approach. So maybe you use a wide field fluorescence imaging and you say, this area is low resolution, but it looks suspicious. Then we use our high resolution technique and go there. So that's one way of reducing your sampling, or the areas that you need to sample. The other area is the brute force approach, where you do spend the time to sample every area. You have to be fast. You have to couple that with algorithms that can tell you if it's positive or negative, good or bad. And then the final one is to have some sort of apparatus that would change your field of view. So maybe use your same optics, but you scan large areas at low resolution, or you undersample, and then you find areas that look suspicious, and you begin to increase your sampling interval over that. So those are the three mechanisms. And they still, we've been kind of looking at all three of those, but I think all of them really could benefit from improving our ability to sample. So first of all, sampling high enough, fast enough, then having that algorithm, the code, the algorithm, the fast analysis to classify that data, to use databases, use other biomarkers to say what type of tissue is that, to be able to then display in real time the image data, display the assessment or classification, provide that information in real time back to the physician or surgeon, and say this is what we think this is, or at least it's suspicious, go back and look at it. And there are precedents for this, because if we look at just pap smears, which basically are collecting sampling from the cervix to look for possible dysplastic or cervical cancer, these are so common that they have automated routines where you would send slides into this. They would identify, do basically image analysis, and identify suspicious cells then that the pathologist can then take a look at and really be able to simplify the time without having to look at all those slides. So that sort of paradigm exists in medicine. Yes? None. Yeah. Yeah. So the question is, could I mention other efforts of parallelization in my lab? And we've been so focused on really the technology and the imaging technology, focused on trying to find what is the best applications for this, looking at all different tissue types and applications. We really haven't spent a lot of time thinking about the processing. And what's possible. Like I said, up until very recently, we weren't limited by processing. We were limited by the optics. But just in the last couple of years, that's flipped. And so now we need to spend time to think about the processing. I think that I think there's just kind of many areas, and obviously from the speakers you've been having this semester for this series, this is just one example of medical imaging in optics, optical biomedical imaging, but MRI, ultrasound, all these modalities I think can really benefit from that parallelization.
Thank you very much.
Okay, thank you again. Thanks.